Just a quick review on our genetic engineering model, just to refresh your memory. So everyone had at their table this little kit made by yours truly. And it has a key, as you can see, with all of the parts and pieces and what they are, and then all of these wonderful parts and pieces. So let's just review what the heck they are. It's been a while, I know. So these are called plasmids, and these are circular pieces of bacterial DNA. And we ordered from Miramar 2 plasmids. The first one has the very, very important red fluorescent protein gene, and that's going to actually make a red fluorescent protein. And we stole that gene from a sea anemone that can glow red. This is a promoter gene right here, this blue bead, and that one basically just triggers the red fluorescent protein gene to jump into action and make the red fluorescent protein. And the third gene on this plasmid was this kanamycin resistance gene, which is a pretty amazing thing. It's a gene that basically allows a bacteria that has this plasmid will not die in the presence of the antibiotic kanamycin. And so it really makes a very strong bacteria that can get this kanamycin resistance gene, because you can't kill it with kanamycin. Now, the second plasmid also had a resistance gene. This one was to the antibiotic ampicillin, which many of you have probably been on for ear infections or strep throat or that kind of thing. It also has right here, this is an arabinose C gene, and basically it's going to allow the bacteria that gets this specific gene to turn on the promoter and the red fluorescent protein gene. So it kind of causes like a little chain reaction. So if we plate our bacteria in a solution that includes arabinose sugar, then we're going to get that bacteria to make the red fluorescent protein. And finally, right here, we have a promoter gene, and that's just going to basically help with DNA replication, which is an important step in the process of getting more of the plasmid made so the bacteria again can make the red fluorescent protein. All right, so we kind of modeled the steps of the lab. First of all, we decided that this plasmid by itself nor this plasmid by itself is enough to actually get a bacteria to make the red fluorescent protein. And so we had to model actually cutting these plasmids apart and then gluing them back together to find the exact right combination of the plasmid we needed. So to do this, we used restriction enzymes, and they're just represented here by little scissors, so BAM and Hindi. And these restriction enzymes cut at very specific DNA sequences. And we modeled this by simply just untwisting our pipe cleaner, because that's pretty much all that's happening, is the DNA is just being cut like I'm modeling here. And so we just at the end of this process ended up with 4 pieces of DNA. As you can see here. So 2 white pipe cleaner pieces and 2 blue pipe cleaner pieces. Okay, so that was going to be called the restriction digest. The second stage was then called ligation, and that's where we took another enzyme. So the restriction enzymes are no longer in the play, but now we have an enzyme called ligase, and we used little clips to represent ligase, and we started just dumping the ligase and all of these 4 pieces together, and overnight ligase was busy running around and basically causing the plasmid fragments to reattach to each other, because remember that the restriction enzymes made something called a sticky end. So it's a little part of DNA that sticks out where it doesn't have its base pair attached to the other side of it. And so if you add something that's going to cause DNA to stick back together again, then all the little sticky ends will run around trying to find other complementary base sections to stick to. So you ended up with 10 different combinations, and I'll let you figure out how exactly that worked. But basically, you could have 2 of these small white plasmid chunks together, 2 big white plasmid chunks together, 2 big blue, 2 small blue, and then all the combinations. So you ended up being able to make 10 different combinations out of those 4 parts. But this is the only plasmid we really care about. This is the one that has all of the 5 genes necessary to make the red fluorescent protein. So the only one we basically left out was the kanamycin resistance. Okay, we only need one resistance gene to be able to select for that particular plasmid. Then the next thing we modeled is we modeled what was called transformation. So we made this plasmid using restriction, digest, and ligation, and then we want the bacteria to actually take in this plasmid. So inside the bacteria goes the plasmid, and we did this by heat shocking and soaking in some calcium ions, and that allowed that membrane to be porous enough for the plasmid to enter. So we kind of said transformation is beep, 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 bam. Now this lucky bacteria is ready to be plated on some agar. And we were going to have 3 different petri dishes. So the first petri dish just is going to have luria broth, which is just agar basically with some nutrients that grows bacteria. We have this plate because we just want to make sure that our bacteria are alive, that we actually have some competent cells. Then our second plate was luria broth plus ampicillin, so a nasty antibiotic that can kill bacteria. And so this was our way of testing to see if we got any transformed bacteria. Because if there was bacteria that grew on this plate, then that means they had to get the ampicillin resistance gene. Okay? And then finally we had a third plate. And again, luria broth, food, ampicillin to kill off anything that's not transformed, and then arabinose sugar. And this is the cool little trigger mechanism that's going to now allow the bacteria to start making the red fluorescent protein. So we went ahead and put those in the incubator and we let magic happen. And what we hoped was going to happen overnight is that this red fluorescent protein gene was going to be turned on, and then transcription and translation of our DNA was going to occur. And transcription, remember, is just the copying of the DNA so that you get a copy out onto the ribosome, and then that little workbench is the place where the amino acids can then be assembled to make our actual protein. And remember, the protein we were really dying to make was this red fluorescent protein. We can do this with making insulin. There's other medications that we can trick bacteria into making for us. And then our final step, of course, is you have to get this specific protein out of the bacteria to make it into drug form, or get it out and do whatever you're going to do with it. And that's a protein purification step. And that involves some chromatography columns and salts, and you're washing out all the different proteins, but only this specific protein was going to stick to one of the salts. So we were going to actually be able to isolate this protein out. If it had been a drug, we could encapsulate it and we could, give it to people. In our case, we just wanted to have something beautiful and pink to put under our fluorescing light to see how beautiful it was. So that was our goal for the whole process. I hope that little review helped a bit.